The endomembrane system is actually a network of organelles inside of a eukaryotic cell, a network that begins at the nucleus and ends at the cell membrane. The reason that these organelles are associated with each other, in some cases actually connected to each other, is because they all play a role in one of the most important things that happens in a cell, which is the production of protein. So the endomembrane system functions to move molecules, particularly proteins, along through the different organelles and actually into and out of the cell itself. So this system starts with the nucleus and then goes to the endoplasmic reticulum, particularly the rough endoplasmic reticulum, then an organelle called the Golgi Apparatus, then an organelle called the lysosome, an organelle called a transport vesicle, and the cell membrane or the plasma membrane. The nucleus and the endoplasmic reticulum are actually physically connected to each other. The rest of the endomembrane system is in separate compartments, but they connect with each other through these transport vesicles. And I'll show you what I mean by that in just a minute. Here is a really nice drawing of the endomembrane system. So we're looking at a eukaryotic cell, only a portion of it. Here's the cell membrane in that bright yellow color. Everything over here in red, that's outside of the cell. Everything in orange here, that's the cytoplasm of the cell. If we were going to draw this whole cell, it would go outside of our view. It would go beyond the edges of this slide. Up at the top here, we're shown a portion of the nucleus. You can see the nuclear envelope in this drawing, and the artist has done a good job showing us that the inner and outer parts of the envelope are connected to each other at these structures called pores. Then we can see actually physically connected to the outer membrane, the rough endoplasmic reticulum. Look at all those ribosomes on the outside of that structure. Physically connected to the rough endoplasmic reticulum is the smooth endoplasmic reticulum, very tubular looking. And then a physically separate structure in blue is the Golgi apparatus. And then we also see these little tiny organelles scattered around. These would include the lysosomes and the transport vesicles. And finally, we have the plasma membrane or the cell membrane. So all of these are parts of the endomembrane system. Now, let's talk about how things move inside the endomembrane system. Because again, not all of these structures are physically connected to each other. When proteins are produced and move through the rough endoplasmic reticulum, the next step for the proteins is the Golgi apparatus. But these structures aren't physically connected to each other. So how do we get protein from here to here? The answer is we build a transport vesicle. Transport vesicles are kind of like taxicabs, or I guess Ubers would be a more modern example. What happens when the protein has passed through the RER and is ready to go to the Golgi apparatus is that a piece of the membrane of the rough endoplasmic reticulum is going to bud off. It's going to come off of the RER and create a little compartment called a transport vesicle with the protein inside. That transport vesicle will then come over to the Golgi, and the membrane of the transport vesicle will merge with the membrane of the Golgi apparatus, and the protein will be passed into the Golgi apparatus. Now, once the protein has passed through the Golgi apparatus, which is also a big network of flattened membrane sacs, once it's ready to leave the Golgi, the same thing happens. A piece of the Golgi membrane buds off and creates a new transport vesicle with the protein inside. Now, this protein has a destination. It's going to go somewhere. It's either going to go somewhere back into the cell, which is what they're showing us here, or it might be destined to leave the cell, to be secreted or excreted from the cell. And that's what they're showing us here. The transport vesicles that leave the Golgi apparatus can actually fuse with the cell membrane and release the protein to the outside of the cell. The same is also true. Things that need to come into the cell can actually come in through a budding process that creates a transport vesicle that can then merge into the Golgi apparatus, or perhaps up here to the RER. All kinds of budding and merging goes on with the endoplasmic reticulum and the Golgi apparatus and the membrane. Now remember, lysosomes are also part of the endomembrane system, and lysosomes are best thought of as organelles that digest materials that the cell doesn't need anymore. They break materials down and recycle them. So if something is coming into the cell and forming one of these transport vesicles, sometimes these become lysosomes. They mature and become a new organelle, a lysosome, so that this material that's coming in can be digested and sent out through the rest of the cell. So the Golgi apparatus is part of the endomembrane system. It's a single membrane organelle. It's shaped into these flattened sacs, similar to the endoplasmic reticulum, but there are no ribosomes on the Golgi apparatus, and the Golgi apparatus has a particular orientation in the cell. In other words, it has what we call directionality. We give the Golgi apparatus names to both of its sides, if you will. There's a side to that organelle that faces the endoplasmic reticulum, and the other side of the organelle faces the plasma membrane. So we call the side that faces the rough endoplasmic reticulum the cis face, and we call the side that faces the membrane the trans face. This directionality allows us to talk about the Golgi apparatus and discuss it among ourselves with different scientists and know which side of the organelle we're talking about. The Golgi apparatus has the job of processing and packaging protein. Particularly, what it's doing is adding bits and pieces of carbohydrate to the protein. A lot of the proteins, particularly proteins that end up on the membrane of a cell, have little pieces of carbohydrate added to them. When you add a little bit of carbohydrate to a protein, you create a molecule called a glycoprotein. And of course, the Golgi apparatus is part of the endomembrane system. On this slide, we have a drawing of the Golgi apparatus on the left, and we have an EM image specifically a transmission EM image of the Golgi apparatus on the right. If you look here on the left, you can see that there are these flat sacs of membrane stacked on top of each other. The ones in the center of the Golgi are the widest, and then the ones on either side are more narrow, shorter, if you will. The side of the Golgi that faces the rough endoplasmic reticulum is called the cis face. This is sort of the receiving side, if you will. Proteins are going to come out of the rough endoplasmic reticulum, go into transport vesicles, and come into the Golgi apparatus on the cis face. They'll then travel through the Golgi apparatus and come out of the Golgi apparatus through the trans face. So this is the exiting side, or the shipping side. The proteins are going to be shipped out through the trans face. If you look at the EM image, the thing that sort of jumps out at you are all these little white circles. These are all transport vesicles, traveling around, joining in with other pieces of membrane and other organelles in different parts of the cell. You can't see it, but the rough endoplasmic reticulum is up here. So that means this side of the Golgi apparatus, which right now is covered with these transport vesicles, this is the cis face, or the cis side of the organelle. Things are entering the Golgi up here. Down here, where there's lots of other transport vesicles, this is the side where things are exiting. This is the trans side of the organelle. Lysosomes are very simplistic in their structure. They are just circular organelles surrounded with a single membrane. So if you were looking at a cell at any moment in time, you would not be able to tell the difference between a transport vesicle and a lysosome. They would look identical, just circular compartments surrounded by a single layer of membrane. The function of the lysosome, though, is to digest whatever material gets inside of it and then recycle the molecules that are produced through that digestion process. So a piece of protein, for example, would be digested down into individual amino acids. A piece of membrane might be digested down into individual phospholipids, and so on. Lysosomes are capable of breaking down virtually all biological materials. And they will break those materials down into their most basic subunits, their most basic components, so they can be used again, so they can be recycled. Over here on the right-hand side of the slide, you can see a transmission electron microscope image of a eukaryotic cell. We're just looking at one particular little spot in the cell. And these dark colored structures here, these are lysosomes. You can also see around the lysosomes sections through mitochondria. All of these circular structures that look striped, those are mitochondria. The other circular organelle that we need to be familiar with is called a peroxisome. Now, a peroxisome has some similarity to a lysosome in that its job is going to be to break some things down. The difference is that the peroxisome has a very short list of things that it's going to degrade for the cell. Peroxisomes have a single layer of membrane around them, just like the lysosome, just like the transport vesicle, but they're going to degrade molecules that a lysosome can't break down. For example, Peroxisomes are able to degrade a molecule called hydrogen peroxide, H2O2. Hydrogen peroxide is formed in living cells as a result of the fact that oxygen gets metabolized, and it sometimes takes on forms that are toxic to the cell, including the form of hydrogen peroxide. So the cell must degrade this molecule when it forms, and it does that in the peroxisome. The other thing that the peroxisome can degrade are things like alcohols, including methanol and ethanol, and even chemicals like formaldehyde. So there are some chemicals, even naturally occurring chemicals, that lysosomes just don't have the enzymes necessary to break apart. And those molecules are dealt with in the peroxisome. You can imagine that something like a liver cell in a human or another animal is loaded with peroxisomes, because the liver is one of the organs in the body that does a lot of the job of breaking down molecules. Over here on the right, you're looking at another transmission electron micrograph image of a eukaryotic cell, a portion of a eukaryotic cell. They're showing us that these linear structures here that are stacked up, and over here, these are Golgi Apparatus. There's all kinds of membrane scattered around here. Some of this is likely Endoplasmic Reticulum. There's probably mitochondria in here in some of these circles. But this big structure right here, this is a peroxisome. Again, you would not be able to look through a microscope, a light microscope or any kind of microscope, and tell the difference between a lysosome, a peroxisome, or a transport vesicle. They're all circular organelles.